Amino acid metabolism is one of those topics that feels overwhelming, so many pathways, so many enzymes, but what if you could recognize the patterns instead of memorizing every step? For the MCAT, you don't need every detail. You need the high-yield logic behind how amino acids are broken down, converted, and used for energy. In this video, we'll simplify transamination, deamination, and key metabolic fates so you can quickly spot what matters on test day and turn confusion into confidence. Transamination and deamination are the key processes that remove amino groups from amino acids for metabolism. When amino acids are broken down, the body must first deal with the nitrogen-containing amino group, and this is where transamination comes in. In transamination, the amino group is transferred from an amino acid to an alpha-keto acid, most commonly alpha-ketoglutarate, forming glutamate. This reaction is catalyzed by aminotransferases and requires vitamin B6 as a cofactor, which is a classic MCAT detail. Think of transamination as simply swapping functional groups without releasing free ammonia yet. Then comes oxidative deamination, primarily in the liver, where glutamate loses its amino group as free ammonia through the action of glutamate dehydrogenase. This step is important because free ammonia is toxic, especially to the brain, so the body quickly converts it into urea for safe excretion. On the MCAT, you might be asked to identify which step produces ammonia directly or which vitamin deficiency would impair amino acid metabolism, pointing you toward B6. Also, remember the connection to the citric acid cycle. The carbon skeletons left behind after transamination can enter energy pathways, linking amino acid metabolism to overall energy production. The urea cycle converts toxic ammonia into urea for safe excretion. Once ammonia is generated from deamination, it cannot accumulate because it disrupts pH and neural function, so the liver rapidly channels it into the urea cycle. This cycle begins in the mitochondria with carbamoyl phosphate formation, combining ammonia with carbon dioxide, and then proceeds through intermediates like ornithine, citrulline, argininosuccinate, and arginine before producing urea. A helpful mnemonic to remember the order is, ordinarily, careless crappers are also frivolous about urination, representing ornithine, carbamoyl phosphate, citrulline, argininosuccinate, arginine, and urea. The produced urea is then transported to the kidneys for excretion. On the MCAT, you may see questions about hyperammonemia, which occurs when this cycle is impaired, leading to neurological symptoms. It is also important to recognize that the urea cycle is energetically costly, using ATP, and is tightly linked to amino acid catabolism in the liver. This concept also connects to nitrogen balance and liver function, both of which are common themes in passage-based questions. Amino acids are classified as glucogenic, ketogenic, or both based on their metabolic end products. After the amino group is removed, the remaining carbon skeletons can enter different metabolic pathways. Glucogenic amino acids are those whose carbon skeletons can be converted into glucose through gluconeogenesis, typically by forming intermediates like pyruvate or citric acid cycle components such as oxaloacetate. Ketogenic amino acids, on the other hand, are converted into acetyl-CoA or acetoacetate, which can be used to form ketone bodies but cannot be used to produce net glucose. A key MCAT fact is that leucine and lysine are purely ketogenic, and a common mnemonic is leucine and lysine are purely ketogenic. Some amino acids, like isoleucine, phenylalanine, and tyrosine, are both glucogenic and ketogenic. This classification becomes especially relevant during fasting states, where the body relies on gluconeogenesis and ketone production for energy. On test day, you may be asked to identify which amino acids contribute to blood glucose or ketone body formation, or how metabolism shifts during starvation. Carbon skeletons of amino acids enter central metabolic pathways like glycolysis and the citric acid cycle. Once the amino group is removed, what remains is essentially a carbon backbone that can be repurposed for energy production. Depending on the amino acid, these carbon skeletons can become pyruvate, acetyl-CoA, or intermediates such as succinyl-CoA, fumarate, or oxaloacetate. This is why amino acids are considered a flexible fuel source, especially during prolonged fasting or starvation. For example, alanine can be converted into pyruvate, linking amino acid metabolism directly to glycolysis and gluconeogenesis. On the MCAT, this often shows up in questions about metabolic integration, where you must track how molecules move between pathways. It also ties into the concept of anaplerotic reactions, which replenish citric acid cycle intermediates. Understanding these entry points helps you reason through complex passages where multiple pathways intersect, especially in tissues like liver and muscle. Amino acid metabolism is tightly regulated and increases during fasting or starvation to support energy needs. Under normal fed conditions, amino acids are primarily used for protein synthesis and maintenance, but when glucose availability drops, the body shifts its priorities. During fasting, muscle proteins are broken down to release amino acids, which are transported to the liver for gluconeogenesis and energy production. Hormones play a major role here. Glucagon and cortisol promote protein breakdown and amino acid utilization, while insulin inhibits these processes. A classic example is the alanine cycle, where alanine carries nitrogen and carbon from muscle to liver, contributing to glucose production. On the MCAT, you may encounter questions about hormonal regulation, metabolic states, or how different tissues cooperate during starvation. This concept also connects to broader themes like energy, homeostasis, and the integration of carbohydrate, lipid, and protein metabolism, making it a high-yield area for passage-based reasoning.